Hello world, I'm a molecular biologist and geneticist. In this video, I will talk about my proposal for a home test kit to test COVID-19. Think about pregnancy tests and blood sugar level tests. A pregnancy test tells whether a person is pregnant or not, and it is pretty easy to use. With a blood sugar level test, a person can conveniently use the test to see if their blood sugar level is high or low without going to a hospital. The aim of this test is similar. It is for people to test themselves without going outside. I have contacted multiple people regarding this test, but I did not hear from them, probably because they are already busy fighting with the virus. With this video, I aim to get attention to the method I will explain. I will not monetize the video and will not ask for subscription. I only ask you to like and share the video so the word goes around until it reaches to researchers. Now, let's get to the introduction part. COVID-19 is a pandemic spreading all over the world rapidly since December 2019. By the end of January, there were around 10,000 confirmed cases of COVID-19. This increased to 85,000 by the end of February and 750,000 by the end of March. As of writing this script, there are more than 2.5 million cases. The common symptoms of COVID-19 are fever, cough, sore throat, shortness of breath, and pneumonia. The virus can spread during the incubation period, which is the time when the virus is present in the body of the infected person, but the person doesn't develop the symptoms. The maximum period of incubation is 14 days. This means that a person that carries the virus can spread it to other people without having the symptoms for 14 days. In fact, a report demonstrated that 78% of new cases that have been found on the day of the report did not have any symptoms. Other coronaviruses that infect humans, such as SARS and MERS, can spread through droplets, direct contact, and indirect contact via surfaces of objects carrying the virus. A study that analyzed the spread of other human coronaviruses through indirect contact concluded that these viruses could stay on the metal, glass, and plastic surfaces up to 9 days. This, together with the incubation period, are the reasons why it is recommended for people to stay at their homes even if they don't feel to have the disease. For the same reasons, going to hospitals for tests is risky, but because there is no alternative, it is necessary. This proposal is made to develop home tests to address this issue. To outline the test, a DNA fragment will be synthesized to target viral RNA. Then, a fluorescent probe will be attached to the DNA. This probe will stay inactive as long as it is attached to the DNA. It will be active when it is released. To do so, a restriction enzyme called SAU3AI will be used to free this fluorescent probe when DNA and RNA hybridize. The presence of the virus will be determined with the fluorescence of the probe with the naked eye. The rest of the video is about the technical details of the method. 4 things are needed to be checked to develop the test. First, can we isolate RNA from saliva with household items? Second, can we prepare the DNA fluorescent dye conjugate that is proposed? Third, can SAU3AI free the fluorescent dye? And fourth, will a sufficient amount of fluorescent dye be released? Some plentiful videos and blogs describe how to extract DNA at home. The first step is to gargle salt water to gather cells from the mouth and filling the cup with the salt water that contains the cells. Then, 1 or 2 drops of soap are dropped into the cup to disrupt the cellular and nuclear membranes. To visualize the process, alcohol is mixed with food coloring in another cup, and then the mixture is poured slowly on top of the salt water. After a couple of minutes, 2 layers will be formed. The top colorless layer supposedly has the DNA. The extraction of DNA seems plausible with this method. The same method might also be used to extract RNA because alcohol can also precipitate RNA. There is no need for purification of the RNA. As long as there is RNA in the mixture can be used in a later step. Oligodeoxyribonucleotide phosphoramidates have been used for many applications, including synthesis of nucleic acids and the addition of modified nucleotides to RNA or DNA. In this proposal, they will be used to attach a fluorescent probe to the DNA sequence that will be used to target viral RNA. Firstly, the DNA will be dephosphorylated at the 5' end. This can be done with a commercially available phosphatase enzyme. Then, 2-cyanoethyl N,N-diisopropyl chlorophosphoramidate will be added to the dephosphorylated DNA as described by Nelson et al. As a result, phosphoramidate will be added to the 5' end of the DNA. After that, a fluorescent probe will be added. As an example, the addition of resorphin is shown. Any other fluorescent probe that has a hydroxyl group, such as fluorescein, can be added with the same method. In fact, multiple probes can be checked to determine their fluorescent intensity, and then the selected probes can be tested for the next step. The end result will look like this, and resorphin loses its fluorescence when bound to DNA. SAU3AI is one of the rare endonucleases that can cut DNA-RNA hybrids. The advantage of SAU3AI over the others is that it recognizes GATC on the forward strand and cuts before guanine. On the reverse strand, CTAG is recognized and the sequence after guanine is cut regardless of the following nucleotides. It is highly likely that the fluorescent probes that will be attached after the CTAG sequence will be cut by SAU3AI. If it can cut, it will likely to break the bond that is indicated in the figure. As a result, the fluorescent probe that's been kept inactive by conjugation to the DNA will be released and activated. Some fluorescent probes are visible to the naked eyes, such as resorphin, fluorescein, and coumarin-based probes. These probes are inactive and colorless when they form covalent bonds with other molecules, and when the bond is broken, they gain their active state and have color change. Furthermore, when observed under UV light, they have fluorescence capabilities, but only when they are unbound. Some cheap and high-power UV LEDs are available online with only $1 or $2. However, although SAU3AI might be able to cut the bond between the fluorescent probe and the DNA, the amount of viral RNA gathered through saliva may not be enough to release sufficient probes to be detected without any instrument. For the test in the laboratories, oral swabs are being used. It has been demonstrated that the viral loads of throat swabs reaches the peak at the first week of symptom onset. Moreover, it is likely that the viral load is the highest on the day of the symptom onset. Clinical trials have to be done with the patients who have COVID-19 to be able to determine whether sufficient amount of fluorescent probes are released with this method. Honestly, I have high hopes regarding this method. Although experiments have to be done, the answers to the first three questions are highly likely yes. If this test works, it will be very helpful as the people who have COVID-19 can begin treatment as soon as possible and they won't spread the virus to healthy people by not going outside to test themselves.